All right, so in this last section, we're going to be taking a look at some of the factors that affect the activity of enzymes. So we're going to be taking a look at several of these. The first one is going to be temperature. So what we're going to see is that most enzymes have a temperature at which they function optimally. And for an enzyme that you have in your body, for instance, it makes sense that that would be at body temperature, which is 37 degrees in a human being. And we call that temperature the optimum temperature, right? So the optimum temperature is a temperature at which the enzyme is most active. And in the case of, like we said, an enzyme in your body, that temperature on your graph here would be at 37 degrees Celsius. When you start changing the temperature from that optimum temperature, the activity is going to decrease, right? So we look on our little graph here, right? We have the reaction rate, how effective an enzyme is plotted on the y-axis versus the temperature increasing on the x-axis, right? So we have our optimum temperature where the graph is the highest, right? That is at 37 degrees. As you start increasing the temperature, right, you start affecting now the shape of the enzyme, you start affecting the R group interactions, which would lead to the enzyme becoming denatured, and as a result, on either side of our peak here, right, the activity decreases. It becomes less effective, in other words. All right, so maximum enzyme activity occurs at the optimum temperature. Enzyme activity can also be affected by the pH, right, or the acid or base level in the environment. Just like with the temperature, enzymes are going to be most active not just at an optimum temperature, but also at an optimum pH. And this optimum pH is not something that is true for all enzymes, even the enzymes that reside in your body. This is going to be determined by the enzyme itself and where it's actually found. Right, so this is determined by the enzyme's environment. So for instance, if you have an enzyme that is residing in the bloodstream, right, so for instance, enzyme in the blood, and as you know, the blood pH is around 7.4, right? Well, then the optimum pH for that enzyme should be about 7.4. And as you change the pH, if it becomes more basic, right, the pH goes up, or if it becomes more acidic, the pH goes down. Well, then what happens is the enzyme activity decreases tremendously. And what you're actually doing there is you're also denaturing the enzyme, right? So the enzyme can be denatured, meaning we're affecting the structure of the enzyme at pHs other than the optimum pH. All right, so like I said, the optimum pH for an enzyme is determined by that enzyme and where it's found in the body. So we mentioned if you have one in the bloodstream, its optimum pH is going to be the pH of the blood. But you can see here in this list of enzymes that the pH can vary drastically. The very first one is a good example of that. Pepsin is an enzyme that you find in the stomach, and pepsin breaks down peptide bonds. And because it's found in the stomach, right, the optimum pH for that enzyme would be the pH of the stomach acid, which is really, really acidic, right, between 1.5 and 2. And if you compare that to an enzyme like arginase, which you find in the liver, right? So arginase would tell you it's an enzyme that breaks down arginine, right? And that pH, the optimum pH for this particular enzyme is on the basic side of things, right? So it's moderately basic, about 9.7. So if you compare these two, right, pepsin and arginase, their optimum pHs are drastically different because the environment that they're operating in is drastically different as well, and they've evolved to function effectively in their environmental pH conditions. So the other way by which an enzyme activity can be affected is through the use of something known as an inhibitor. And generally what inhibitors are they are substances or molecules typically that reduce the activity of the enzyme, or we say they cause a loss of catalytic activity. So they reduce or eliminate altogether the activity of in other words, the enzyme is not going to work if you have these inhibitors present. And the way that they work is actually very simple. What they do is they prevent the enzyme from doing what it's supposed to do, which is to work on a substrate, to either break something down or build something up. So the way that these inhibitors are going to work is they're going to prevent the substrate from fitting or from binding to the enzyme active site. All right, so normally what we saw before is that your enzyme and your substrate can combine, form your enzyme substrate complex, ES. Enzyme does its thing and releases the product. Enzyme is freed up and it can go back and then repeat this process over and over again. Right? So this is what happens typically. When there is an inhibitor present, though, I here now would represent the inhibitor. Right? What can occur now is the inhibitor can bind to the enzyme and form an enzyme inhibitor complex. And as long as that inhibitor stays bound, it prevents the substrate from binding. And if the substrate cannot bind to the enzyme, well, guess what? No product can be made, because the only way you can make a product is if you have your substrate and enzyme bound together. So that's generally how your inhibitors are going to work. We're going to see a couple slightly different ways by which they can accomplish this. The first one is called competitive inhibition. And what a competitive inhibitor does is it competes with the substrate for the active site. And the way that it's able to do this, because remember the active site has a particular shape, a competitive inhibitor then is going to have a shape that is similar to that of the substrate. So in other words, the inhibitor and the substrate resemble each other. In terms of their shapes. So the enzyme can't really tell which is which. Is it binding to an inhibitor or a substrate? Because they both look the same. The way that we can reverse this effect is by simply having more of the substrate there. So we just simply drown out the inhibitor, right? Let's say now, for instance, if we had, we'll just draw a really simple diagram here. If this is my system, I have a box, there's my enzyme. Okay, and there's the active site. It's pretty simple. It's like a circle. And let's say now the red dot here represents the substrate. And then the green dot represents an inhibitor. Right? So if I have one substrate molecule in here and one inhibitor molecule, a green dot, the chances of binding the substrate to the active site is 1 in 2, right? You have a 50% chance of the active site being occupied by the substrate or by the inhibitor. So the way then that we can reduce the chances of the inhibitor binding is simply by having more of the substrate, right? So if we now have a lot more of the red dots, well, then the chances of binding the inhibitor decreases dramatically. All right, so increasing the concentration of the substrate reduces the effect of a competitive inhibitor, right? Just by simply drowning it out, right? And this works because they both resemble each other in terms of their shape, so they're competing for that active site. So because we have way more of one than the other, there's no competition, right? The red substrate here is going to win that battle every single time. All right, so competitive inhibition, there's our active site of our enzyme in both cases, both the substrate. So normally, this is what we would expect to happen on top. So normally, right, your substrate comes in, binds to the active site, forms your enzyme substrate complex, enzyme does its thing, releases the product, and then it can do this over and over again. On the bottom now is what would happen if there's an inhibitor present. So you'll notice that the part of these, or the substrate and the inhibitor, they resemble each other in terms of their shape, right? Similar shapes, specifically the part that would interact with the enzyme active site. So if the inhibitor binds, well then the substrate, right, can't bind to it because it's occupied. So the enzyme isn't able to do what it's supposed to be doing normally, so no product can be made. Now eventually what can happen is the inhibitor here can fall off, and then the substrate, if that enzyme active site is open, the substrate can come in and bind eventually, right? Because it's not a permanent interaction. So that's competitive inhibition, where the substrate and the inhibitor resemble each other in terms of their shape, so they're going to compete for the active site. We can reduce the effect of the inhibitor by simply having more of the substrate present. We can increase the concentration of the substrate to reduce the impact of the inhibitor. The second type of inhibition is known as non-competitive inhibition. And in a non-competitive inhibitor, this is going to have, again, this molecule is going to have a structure that is different than that of the substrate. And the reason why it doesn't need to be the same structure as a substrate is because of what it does. It binds to the enzyme not at the active site. So it binds at a site other than the active site. And what it does is it changes the shape of the enzyme, specifically the shape of the active site, and as a result now the substrate cannot bind to it because the area that it was supposed to go into, it doesn't have that shape anymore. All right, so if we take a look here now, right, so normally on top here, this is what we would expect to happen. Right, so normally we have our enzyme with the active site, substrate can come in, form your enzyme substrate complex, enzyme does its thing, product gets released over here, and then the enzyme can come back in and repeat this process over and over and over again. All right, now what we're saying is, in this non-competitive inhibition, right, this inhibitor can come in and it can bind not at the active site, but maybe binds here. Binds at a location other than the active site. And what this leads to is it changes now the shape of the active site, right? So now instead of being this particular shape here, the optimum shape, now it's changed in this really elongated shape. Obviously, this substrate now cannot come in and bind. So the enzyme isn't able to carry out its function. Because the inhibitor is binding at a place other than the active site, increasing the amount of substrate isn't going to reverse the effect of the inhibitor, right? Because the active site doesn't have that same shape anymore, right? So this is non-competitive inhibition, where the inhibitor binds at a place other than the active site, and it changes the shape of the active site, which prevents the substrate from binding. The last type of inhibition here is called irreversible inhibition. Right, and all of the two previous examples that we talked about were reversible inhibitions, meaning that eventually the inhibitor would fall off and then the enzyme can return to its normal function. In an irreversible inhibitor, the molecule that binds, the inhibitor that binds to the enzyme, causes the enzyme to lose all of its activity, right? So that means it cannot convert substrate to product anymore, and it loses all its activity permanently, right? It means that it cannot be reversed. So for all intents and purposes, an irreversible inhibitor kills that enzyme, right? Which, as you can imagine, is going to be pretty serious. Lots of times these are going to be toxic substances that are going to destroy that enzyme. And one of the main reasons why it's an irreversible inhibitor is because it forms a covalent bond, right? A permanent bond with one of the amino acid R groups that causes a permanent change in the activity and the shape of the enzyme. And that's the way that lots of things work. For instance, your antibiotics that you would take to fight off a bacterial infection, right? The way that they work is they actually permanently deactivate the enzymes that you would find in your bacteria, right? And humans don't have bacterial enzymes, so it doesn't affect us, but it does affect the bacteria. So that's the idea behind those types of inhibitors. And probably one of the more common ones that you may be aware of are penicillin antibiotics and different types of penicillins, right? So this is the basic structure for a penicillin molecule, and different types of penicillins. Usually your drugs that end in cillins are all based on this structure. The difference between them is what this R group is, right? So you may have heard of ampicillin or amoxicillin, right? If you just stick this in place of this R group, that makes it ampicillin. If you change it slightly, you attach an OH to the benzene ring here, and you place it instead of that R group, then you have amoxicillin, right? So what pharmacists do and people in organic chemistry do is they're constantly on this never-ending battle with trying to outwit your bacteria, and by changing what that R group is, right, you can subject the bacteria to different types of antibiotics. So they, if they do develop resistance to one, we can, modify it slightly, and that way they're not resistant to the new one. All right, so these all work by irreversible inhibition of those particular bacterial enzymes.